for the recent Casp. Okay, so let's try that again. We're participating in the Casp17. Here's a puzzle. This is a real drug-like molecule, and this is a real human protein. We know the two of them to bind together. There's just one catch. The protein has 3 different pockets the molecules could sit in, and they're scattered right across it. So which one is real? That single question is the prediction. Let's work it out. This is for the Casp17 competition, and this is target 2383, T2383, and we're trying to figure out what pocket it actually binds in. Here's the protein we're looking at. First, meet the protein, because DDB1 is generally one of the most remarkable machines in your cells. Think of it as a universal adapter for a molecular shredder. DDB1 sits at the heart of an enzyme that tags other proteins for destruction, and it's modular. Snap in one of the dozens of interchangeable receptor parts, and the same machine now destroys a completely different target. One adapter, a whole family of shredders. And DDB1 is a hub. There's a whole family of ligases, and we can snap in one of the interchangeable DCAF receptors, and DDB1 becomes a different CRL4, each destroying a different target protein. And it was first discovered as a DNA damage sensor paired with DDB2. It patrols the genome and flags UV lesions for repair. And it's been hijacked by cancer drugs, so we can use it for helping to treat cancer. And it's also used by viruses to invade the cell and wreck the cell's defenses. And we can see that molecule here. Here's a live view of the protein, and you can see there's all the different beta barrels. And you can even see right through them, so that you can see directly through all these things in this giant molecular machine. Here, let's just reset this. Great. DDB1 is a molecular adapter, the hub of the CRL4E3 ubiquitin ligase. It helps decide what proteins the cell destroys. 3 interwoven WD40 beta propellers. BPA, BPB, BBC, plus a C terminal cap. And that's shown here with the blue, green, and orange. You can see that here. The 3 different domains. WD40 beta propellers. And this one is kind of here. And then the pink is the C terminal cap. So the blue is BPA, The green is BPB, the orange is BPC, and the magenta is the C-terminal cap. And this is a prime molecular glue target. A small molecule here can reprogram degradation. The ligand is a off-the-shelf molecule, and it's enamine Z95-430-1282 with 25 heavy atoms. You can see a picture of it here. It's a benzyl oxadiazole tied to a chloral trifluoral methyl pyridinone, two flat aromatics, a halogen tail. There's no prior data for this target, so they just pulled it out. And you can see that over here. Giant name, 1,5-benzyl, 1,3-oxadiazole, 2-il-methyl, 3-chloral, 5-trifluoromethyl, pyridine, 2,1. And we can see that here. Let's just view that. Here's the molecule. The fluorines are very interesting. And it has 3 validated pockets, and they sit 33 to 55 angstroms apart. There's BPA, the fragment site, C-terminal binder, micromolar binder, del BPC nanomolar for a new ligand. Which one is real? That's the entire prediction. So here we go. Here we have the different binding sites we found. 3 main binding sites, 3 main pockets. However, the methods disagree. The learn scores, GINA, GenScore, and the LLM choose del BPC. And the physics with 58 nanoseconds molecular dynamics and molecular mechanics, that chose the C-term and BPA. And the structural prior, the blind cofolds chose BPA. The molecular dynamics even redefined the known C-term binding residues from scratch, a pipeline self-check. Only BPA survives every method's shortlist. So we hedge 5 models, 5 sites. We weight physics over the learn scores and lead with the C-terminal pose, but we place a model at every credible site, anti-catastrophe insurance, and the Casp scores all 5 regardless of order. And it's all blind for now. When it drops, we score all 5. Learn score versus physics decided on a real multi-pocket target. And I'm Stephen Ness from Dioida Bio. Datasets for AI and humans. Check, check. Check 1, check 2, check, check. Check 1, check. Hi, I'm Stephen Ness, and we just are participating in the Casp17 Protein Ligand Docking Challenge. We worked on. Hi, I'm Dr. Stephen Ness, and we're talking about the Casp17 Docking Challenge. We're working on the protein ligand docking, and we're looking at target T2383, and we're trying to figure out which pocket does the drug actually bind. We're looking at human DDB1 and a generic small molecule with 3 plausible homes. DDB1 is a hub. There's a whole family of ligases that snap into dozens of interchangeable DCAF receptors, and DDB1 becomes a different CRL4, and each one destroys a different target protein. And we can take a little look at it over here. Here's an electrostatic view of the surface of the protein. One thing is interesting, you can see right down these beta propellers right through. It's a DNA damage sensor paired with DDB2. It patrols the genome and flags UV lesions for repair. It's hijacked by cancer drugs, so we use it as a cancer drug to disrupt this and degrade what we want to, and also by viruses. It's a molecular adapter. It's the hub of the CRL4 E3 ubiquitin ligase and helps decide which proteins the cell destroys. It has 3 interwoven WD-40 beta propellers. You can see that here. There's blue is the BPA, green is the BPB, orange is the BPC, and then purple is the C-terminal cap. And it's a prime molecular glue target. A small molecule here can reprogram degradation. It's an off-the-shelf molecule. Here's the chemical structure. It has some interesting fluorines and a chlorine, lots of rings, a benzyl oxidazole tied to a chloral trifluoromethyl pyridinone with 2 flat aromatics and a halogen tail. Has 0 recorded bioactivity, so we don't know anything about this in terms of working with proteins. We can see a view of it here. There's a 3D view of the ligand molecule. view. You can see the fluorines here. There's 3 validated pockets from previous crystal studies, the BPA, C-terminal, DEL, BPC, and we're trying to find which one of these pockets, or is it in an even new pocket that's unknown? And we can see that here. We see the 3 molecules from the 3 crystal structures in orange, green, and blue. The methods disagree. The learned scorers choose DEL BPC, the physics term choose the C term or BPA, and the structural prior, the blind cofolds chooses BPA, and the MD even rederived the known C-term binding residues from scratch, a pipeline self-check. Only BPA survives every method's shortlist. So we hedge. We make 5 models, 5 sites. We weight physics over the learn scores and lead with the C-terminal pose. But we place the model at every credible site for anti-catastrophe insurance, and the Casp scores all 5 regardless of order. Here's our 5 predictions that we made. And this is all live with Smol, so you can see this here. Like we could show the surfaces. These are all the different proteins. And it's blind for now. We have to wait for the competition to be over. And when it drops, we score all 5.